The name of the speaker in the last session of day 3 is Dr. Subhan Kumar Sahu, Associate Professor, Sardar Vallabhbhai National Institute of Technology, Surat. So, title of his talk: Fluorescent Nanoclusters and Application. So, Dr. Sahu is currently working as a associate professor in the Department of Chemistry, Sardar Vallabhbhai National Institute of Technology, Surat, India. He has more than 18 years of teaching and research experiences. He was awarded twice with Young Scientist Award from Punjab Academy of Sciences in 2004 and Orissa Chemical Society 2009. He worked as a visiting professor at University of Cagliari, Italy, and research professor at Kwangpuk National University, South Korea. He has published more than 190 research articles in the field of sensing and biosensing, nanosensing, supramolecular chemistry, synthetic coordination chemistry, and studies of metal complexes in solution. He has edited a book on sensing and biosensing with optically active nanomaterials published by Elsevier, and it is more than 10 books chapter and 1 Indian patent. Recently, he was considered as 2021 emerging investigator by New Journal of Chemistry, RSC, publisher RSC. So now, sir, you can start. Okay, I share with my. Let me share your slide.
Okay. Thank you for the nice introduction. My voice is clear?
Yes, sir. Yes, sir. It's clear. Sir, is it visible, sir?
Yeah. Make it a presentation mode.
Yeah. Now it is fine.
Yeah. Yeah. Now it is okay. So. Thank you for inviting me to deliver some of my research work, and I hope that you will enjoy some of my outcomes from my laboratories in the field of sensing and the biosensing. Okay, so mainly I will talk about the fluorescent nanoclusters and its application in the field of sensing and biosensing. And mostly the example that I am going to discuss, that is performed from my research group. So next slide, please.
1 minute, sir. It's not.
Okay, no issue.
Is it fine?
So what? Yeah, it is fine, but
okay,
just okay.
Is it changing?
So yeah, it is changing
okay,
now.
Then, sir, okay.
In this talk, I'm going to discuss the fundamental about the fluorescence spectroscopy. We and then nanoclusters and it in the field of sensing and the biosensing. So far you have listened so many good. I will give for the youngster, those who are at the MSc or maybe a little bit at a higher stage, or those who are wish to work on the fluorescence sensing. I will discuss something about the fundamental on the fluorescence spectroscopy. Then I will teach about to how you can design the fluorescence chemosensors by using both organic dyes as well as the optically active nanomaterial. And then my talk will be mostly concentrated on the examples based on the sensing with the fluorescence nanoclusters. Next slide, please. Okay, so generally whenever we are talking about the fluorescence is nothing but a kind of luminescence. So we need to understand what is luminescence. Luminescence is nothing but the emission of the light, but that emission is occurred through different procedures. That, for example, if you react, some chemical reaction will undergoes, and you have seen so many light sticks also. Okay, so due to some chemical reactions, the light will emit. That we call as a chemiluminescence. For like this, when the radioactive elements emit light, that we called as a radioluminescence. When the emission occurred from a insect, that is because of some biochemical reaction, that we called as a bioluminescence. Like this, you have different type of luminescence, for example electroluminescence, your sonoluminescence, including the photoluminescence. In the photoluminescence, you applied a source of energy in the form of light, and fluorescence is nothing but the your um, a kind of fluorescence. Okay, so if you need to understand more, you need to understand about how the fluorescence occurs. Example to understand about the Jablonski diagram. Mostly when the organic fluorophore, including quantum effect, the electron will goes to the and they will come to the ground state, and when they will come to in a steady beta, where the electrons come to the ground state from the S1 state to the ground state, and it releases the energy that we called as a fluorescence. And there are other mechanism also there. Sometimes the electron, when you are exciting excited to the higher energy, it may undergo interconversion, intersystem crossing, and convert to the triplet state. And when the triplet state, the electron come to the ground state, and when it release the energy, that we call as a phosphorescence. But mostly the we are generally discussing about the fluorescence, and we will discuss about the fluorescence. So we need to understand that you need to excite your molecules. with Without proper excitation, you are not going to get the fluorescence. Okay, once excited, the electron goes to the higher energy state. From that higher energy state, it will come to the ground state, and at the ground state, it will release the energy. Okay, so one of the thing is that the energy at which you will excite your molecules, can observe at a lower energy. That means if you are exciting the molecules at 300 nanometers, then your emission you may get after 300, maybe 400, 500, like this. So the difference between the absorption maxima and the emission maxima, that we call as a Stokes shift. This is one of the important parameters when you are working with the fluorescence sensing, biosensing. You need to check that how much the Stokes shift is. And nanomaterial, the fluorescent nanomaterial known for higher Stokes shift, and that's why they have a very wide application in the field of the diagnostics and the imaging. Next slide, please. Okay, so there are some of the factors that we need to understand. One of the, whenever you are working with developing a sensors, so you need to work on, or you have to consider the quantum field. How of sensor is. Okay, the fluorescence may be switch on, switch off, or maybe electric, but what is the quantum field of that? Not in the so how much photon emitted divided by how much photon is absorbed. So experimentally, generally we determine the already known to us. Next slide, please. Next slide, please. Okay, so as I told you, quantum field is one of the most important parameters in developing a fluorescence sensors, and generally experimentally we determine the relative quantum yield by comparing with a known standard fluorophore or whose quantum yield is already known. So in such condition, what you need to do, you have to record the absorption spectra so that the absorption should be lower than your 0.1, so that there will be no self-quenching effect, and you can record the emission spectra of both standard and the your compounds, and then from the emission you find out the area of that emission curve, that you can easily obtain by using a software like Origin, and then you apply equations to find out the quantum yield of the your sensor. So whenever you are developing a sensor, sometimes the fluorescence may be quenched. So reviewer may ask that how how much quantum yield is taking place, or if you are taking a fluorescence intensity, then or if you are taking a probe, then what is the quantum yield of that probe? That may be asked by the reviewer. So it is a very simple way that you can find out the relative quantum yield. And in the fluorescence, there are other factors also very important in the designing of the sensors. Okay, one of the most important thing is the solvent. Okay, so you need to choose the proper solvent, because solvent has a very important effect on your system. Mostly they choose, they first optimize which ratio or which solvent look for appropriate solvent, and whenever you are changing the solvent, the fluorescence property gets changed. So I will show you one of the examples how it changed. Next slide, please. Uh, okay, so you see that here I have shown that how the fluorescence properties of two fluorophore is getting is changed when you are changing the polarity of the solvent. But if you see in that uh, change, the drastic change is occurred when you are changing the solvent from lower to higher polar solvent. The emissions change from the blue to red region, but you see that there is a slight change in the absorption, but there is a drastic change in the emission properties of your fluorophore. So that's why in designing the in the developing of the solvent also, you need to take care and you have to optimize that which solvent, which condition is appropriate for the developing the sensor, including the other factor like pH, also very important for maintaining developing the sensors. Next slide, please. Okay, so in the fluorescence spectroscopy, generally, if you see that your x-axis is wavelength, but y-axis is it is fluorescence intensity AU. It is generally mentioned arbitrary unit, because the intensity of that fluorescence spectra is depend on the so many factors. For example, your concentration, okay, and what is the excitation light you have used, the strength of exciting energy, as well as the your solvent property, what kind of solvent property. So intensity, that's why fluorescence intensity in the y-axis in any instrument, it mentioned as a arbitrary unit. You are recording the fluorescence in spectra, you need to take care of concentration, because you take high concentration of your probe or sensor, there may be a chance of self-quenching because of the collision between the molecules. So you have to concentration, so that you can avoid the your self quenching effect. Okay, self quenching, and you see that in generally we use micromolar concentration or 10 to the power minus 5 molar concentration. These are the ideal, minus 5 to minus 6. These are the ideal concentration. Generally, most of the paper you can see they use the concentration of your probe, and using that concentration they go for the your go for the sensing experiment. Next slide, please. Okay, this is an instrumental part. We need not to discuss much on this part. One thing I want to say that there is a slight difference in the UV visible as well as the fluorescence. In the UV visible, we when the light is passed through the sample, we record at the what is the change in the light at 180-degree directly. But in fluorescence spectroscopy, if you are exciting your sample, you record what is the at 90-degree you are. Okay, what is the light emit from that your substance? Next slide, please. Okay, so these are some of the more internal pictures of the fluorescence spectroscopy, but I just want to show the cuvette. In the fluorescence spectroscopy also, you are using the similar quartz cuvette, but in the fluorescence spectroscopy, when you are recording the spectra, you will see all the four sides are transparent, and the internal volume is almost same, and minimum 2 ml is ideally good to record the spectra. But if your sensor is expensive or your material is expensive, you can use other kind of micro cuvette where you can record the spectra by minimum samples. So that are available. So depending upon your experiment, you can purchase the different kind of cuvettes. Next slide, please. You can okay, next. Okay, so there are so many factors when you are developing your sensors, and you when you are taking and recording the spectra, sometimes you are thinking that why you are not getting the fluorescence. Okay, why there, how much intensity, or you are expecting, you are not getting that much. Or sometime you are, if you are adding something, changing pH, the fluorescence getting quench. So there are so many way that fluorescence intensity of your probe can be can be altered. Okay, so one of the most important thing is the self quenching, and that self quenching is generally occurs if you have taken the high concentration of your probe. So ideally, you have to optimize the concentration of your probe also. So you have to take ideally a dilute solution that will avoid the self collision and non-radiative decay from the excited state. So that you can avoid. And the collisions also, that quenching also sometime occurred because of the fluctuation in the pH or some other impurities like some heavy metals and the halides. So whatever the solvent or water you are using, that you have to take care that, and including the pH. If you are maintain a fixed pH or whatever the pH, that you need to be take care. And other, there are other quenching mechanism also there that you can see. One is the dynamic quenching and another is the starting quenching. And there are so many other effects. This is the inner filter effect, that is at higher concentration, that you have discussed, that at higher concentration, when you excite the sample, you will not get sufficient emission because of the internal collision, and the emitted energy will pass to the another molecules. Generally, we called as a inner filter effect. Next slide. And this is one of the important things, that how the quenching of a fluorophore can occur. And this is the effect which is used for the imaging applications. That is great. Here, the emission of a the first fluorophore, the donor fluorophore emission spectra of the first your overlap with the spectra of the fluorophore. So in the first it will emit, that emitted energy will be absorbed by the another fluorophore, and that fluorophore will be emitted. So this is the one of the things that the number of different variety of sensors developed within the FLET mechanism, especially for designing the ratiometric fluorescent sensors. Next slide, please. Next slide, please. Okay, so now with this short discussion, I just want to say that a simple excited molecule, you have a number of phenomena at the excited state. So whenever you are designing a sensor, you will be excited that there are number of different mechanisms, number of process that is occur in the excited state, which you can apply for the designing of the sensor. Not only you can see that there may be some reaction will take place, some conformational change takes place, electron, photon, energy transfer takes place, and some other excimer formation. So number of mechanisms will be takes place once you excite your molecules. So this makes in designing variety of sensors for multiple applications. So now I'm coming to the my topic, that how you can develop this fluorescence sensor. So now you have understand little bit about that what is fluorescence spectroscopy and how much diversity it has. One of the direct fluorescence spectroscopy is more sensitive compared to your UV visibility. That's why it has widely applied for developing sensor for various application. Here, whenever you are designing a fluorescence sensor, all sensors you need to focus on the three important things. One is the your light emitting unit, second is the binding unit, third is the sensing mechanism. The binding unit will recognize once the target recognize your binding unit recognize target and allied, then it will change the design. You see that first and second. Here in the past, the light emitting unit and binding unit, is separated by some group ethyl or maybe ethyl. Okay, some group may be separated. So you can have a you can design like this, or there is a third type of your fluorescence sensors where once your analyte is interacted with the probe, a new compound is formed, and that new compound will show different fluorescence, that we generally called as a chemodesimeters. Okay, and the fourth type is your indicator displacement approach. This is a supramolecular approach. You can say that here the light emitting unit and the binding unit is non-covalently interact to each other. And a analyte having a higher affinity with the binding site, it will interact with the binding site and it will take away the binding site and release the your light emitting unit, and you can retrieve or recover the fluorescence property of your fluorophore. So mostly this you can design, and there are a number of review that we have presented uh, uh, written by from my groups that you can go through it. The recent one you can read, that is the New Journal of Chemistry, that a invited review that will show you the how we are doing in the field of the sensing field. Next slide, please. uh, Anand sir, next slide, please. Okay, so generally, when you are entering to the sensing field, you can focus on either on the designing the new kind of dyes, or maybe optically active nanomaterial or quantum dots, or that light emitting unit, or you can focus on the sensing mechanism. What kind of sensing mechanism will work? Maybe on FRET, PET, AIE, ICTs. There are so many different mechanisms are there where you can focus. Fourth is the recognition unit. What kind of recognition you take one kind of recognition unit and a number of sensors. For example, in our research group, we are using vitamin B6. Vitamin B6 vitamin B6. You can see these four pictures. This is pyridoxal, pyridoxamine, pyridoxal 5-phosphate. This molecule we are um, uh, develop a number of sensors. And third things, fourth thing, applications. Whenever you develop the sensor, you need to look what kind of application you can do. Depending upon the person, some person do not, some biomedical person, they do not develop, but they apply the probe. So what kind of probe you are, what kind of application, that you can focus on the application also. Somebody will develop the probe and you can look at the application part. And sensing sensor has a wide application, not only in the analytical field, it has a wide applications. So here in the light emitting unit, you have two things. Either you can use organic fluorophore, or you can use the optically active nanomaterial. You can see that using also organic fluorophore, you can design from the blue emitting to the NIR region. So wide variety of organic fluorophore or light emitting units are there, but there are certain advantage. Also, there are some disadvantage also. For example, in the organic system, you have a synthetic difficulty, aqueous solubility. So always, that's why we look for the alternately the optically active nanomaterial also. Next slide, please. Okay, these are some of the examples that we have designed sensor by organic fluorophore. So here in the first example, you can see that pyrene is connected to the pyridoxal. As I told you in the previous slide that we are mainly focusing to use the vitamin B6 cofactor as a binding unit. So here you can see that the pyridoxal is acting as a binding site, and your pyrene is acting as a light emitting unit. When zinc is added to this system, you can see that there is a distinct fluorescent change occurred, that is due to that formation of the hexamer. When this probe makes a coordination with with this probe, the two pyrene molecules stack together, close together, and hexameration led to the gives the your green fluorescence, and that is very that used to distinguish um, other thing, please the other analytical parameters like detection limit, and similarly we have other sensors using decimeter type, the sensor for detection of various organic fluorophore. Next slide, please. Next slide. Next slide, please. Okay, participant, whenever you have any confusion or any doubts or you want to discuss, then you can freely ask me in between. Also, we can discuss. If you have any doubts, or if you are in the field of the sensing or wish to enter into the field of sensing, that just don't hesitate to ask me any kind of questions. Okay. Uh, Sir, can you switch to next slide, please? Any participant, can you reply? Are you here? Can you hear me?
Yes, sir, we can hear you.
Hello. Hello. Sam. Hello. Sir, am I audible?
Yes, sir, you are audible.
Sir is not there now.
Yes, sir, it's not there.
He's disconnected. Sir. Okay, sir. No problem. Okay, sir. Okay. Uh,
Okay, sorry, there is some a problem in the internet. Atta sir, can you share the PPT?
Have you joined? Okay, okay, you already joined, yeah. So now I am sharing a slide.
Okay, up to now my voice is clear. Okay, my voice is clear. Sir, your one is muted.
Yes, now voice is clear, but is it visible? Your PPT
Yeah,
file?
it is clear now. So
Okay, okay.
I think participant, you have heard. Sorry that due to internet problems, some problems are occurred, but no doubt this is the last session. We can continue and we can discuss, no issue. So we have discussed that you can develop a sensors by using organic fluorophore. Okay, but organic fluorophore has its own advantage, including it has own disadvantage also. Sometimes it has a synthetic difficulty, aqueous solubility, which is one of the most important factors in the developing a sensors, and photo stability. So there are so many different kind of problems are there in the organic system. So that's why we are look for the alternate solution where sensor can be work in the pure aqueous medium. The best example, best thing is that we should switch over to the your optically active nanomaterial. Nanomaterial means either it will show some kind of color or maybe some kind of fluorescence optical properties we are showing. So there are variety of nanomaterial which shows intense color or maybe beautiful fluorescence, size dependent fluorescence. And you know that because that surface you can modify different kind of ligand on the surface of the nanomaterials, and you can develop or you can design a number of different kind of sensor within the same nanomaterial. You can coat as you wish what kind of ligand on the surface, or you can go for the post functionalization. Post functionalization means you can link other kind of group on the surface, which surface with the molecule which are already on the coated. So like that you can prepare a number of different kind of sensor by using the same optically nanomaterial. So you need to know about the what is the best choice or what are the choices you have to work on the field of the your nanomaterial. So here is a book that recently it is published by edited by me. Also there are total 17 chapters, and here you can find these books cover the use of the different kind of nanomaterial in the field of the sensing and the biosensing. It contains the quantum dots, semiconductor quantum dots, carbon dots, and different kind of 2D nanosheets that are applied in the field of sensing, which are metal based or metal free nanomaterial. You have a option for polymeric nanoparticles and p dot, which is polymeric dot, you can say organic nanoparticles. You can convert your organic system by co-precipitation method into a organic nanoparticle, and you can apply the sensor in the field of the sensing. I believe that some organic nanoparticle examples may be given by some your speaker in the previous talk. Okay, and there are other options, that is plasmonic nanoparticle, especially that gold and the silver, copper, when their size are 4 to 50 nanometer or above 5 nanometer, we can see that they will show the beautiful color, which you can use for designing of colorimetry sensor. And now in this talk, I will discuss about the fluorescent nanocluster. In the fluorescent nanocluster, what will happen? The same nanomaterial, but they will have the size very ultra small size. Okay, they are gold or there may be silver, maybe copper, but they are of smaller size, so there will be no plasmonic effect. So first I will give some example which you have developed for colorimetry sensor with the plasmonic nanoparticles. Here is the example of the gold nanoparticles. Here you can prepare a gold nanoparticles by using a auric chloride, by your reduction citrate ligands, and then by applying a ligand exchange methods. Okay, you can quote the different type of ligands. And here is the example. You can see that here a pyridoxal derivative was coated on the nanomaterial surface, which showing the red color. So once the metal is added to that your plasmonic solutions, the nanoparticles come closer and undergo aggregations. Due to the aggregation of the nanoparticle, there is a change in the color of that nanoparticles from red to purple, and the plasmonic band shift to the lower energy. So this way you can develop the colorimetry sensor by using the your plasmonic nanoparticle which shows the color. There are other examples that we have performed in our laboratories. This day we have directly coated the pyridoxal and apply for the sensing of the metal ions. Next slide, please. Okay, so these are the, you see that these are the nanoparticles of larger size. That's why they show the color. They will not going to show any kind of fluorescence properties, but they will show the intense absorption, intense color, and their size will be more than 5 nanometers. And you see that the TAM image, the size are close to the more than 10 nanometers, and they will undergo aggregation because of different mechanism, by complexation or because of some kind of non-covalent interactions. The nanoparticle will come closer and color will change, and that color you can use and use for the detection, or you can integrate it with the smartphone. You see that here a smartphone generally people are using widely, that you can use in the field of the analytical field also, because when there is a change in the color, that color from that color change you can record the red, green and blue color intensity using the back camera of your smartphone. In the smartphone you can freely download color coding, various kind of apps are there where it can capture the color intensity, and using the back camera you can develop a calibration curve by recording either maybe blue, maybe green, or maybe red color intensity, or maybe some cases we use the ratio, and you can use the smartphone in the field of the analytical field. So there are a number of publications we have made within the plasmonic nanoparticles. So what I just a brief example I have given, that nanoparticles can be used in the field of the sensing, but these nanoparticles are of larger size. Next slide. But whenever we decrease the size of the nanoparticle to ultra small, that means size smaller than 3 nanometers, less than 3 nanometers, having a few to the 100 atoms of the metal, then it will behave like a your molecules. They have a discrete energy level, and whenever you excite, you can get the electron will go and back, and you will get the your fluorescence, and which generally we call as the fluorescent nanoclusters. And these nanocluster are ultra small, so you will not say that you will not see any kind of properties of plasmonic behavior. Okay, that is collective oscillation of the electrons. that is not, you can, instead of that, because of the smaller size, it will behave like a molecule, and it has a discrete energy level, and that's why depending upon the size of the nanoclusters, you can have a different type of different emission from blue to red emission. These are very exciting, and mostly you can use the metals like gold, silver, copper, and platinum, or maybe a mixed metal, bimetallic or multimetallic metals, using these type of metals for designing these fluorescent nanoclusters. So it has a wide applications, and you see that you can get the, you can design or you can prepare the nanocluster from blue emission to the red emissions, and this is a new kind of your fluorophore, and it is a very alternate to the semiconductor quantum dots or maybe organic dyes. And these nanoclusters shows excellent photoluminescence behavior and photostability and biocompatibility, and you can have a application in the field of the bioimaging, diagnosis, and other kind of applications. Okay, next slide, please. So its synthetic strategy is very simple also. You can take your precursor, the desired metal salt, and a coating ligand, and then mostly coating ligands or some kind of enzymes or polymers or your proteins maybe, and then you can go for the control reduction, or you can go for the etching of the gold larger nanoparticle into smaller nanoparticles. So, but in the field of the sensing, mostly that using the metal salt as a precursor and small ligands as a stabilizing agents are generally used for the synthesis of the your gold nanocluster. So in our group, what we are doing, we are preparing such nanoclusters, and then we are post functionalizing with the vitamin B6 cofactor for the designing of various kind of sensors. Okay, so you can see that depending upon the different kind of ligand that you have used to coat on the surface, you can tune the emission property of your nanoclusters. Next slide, please. Okay, and you see that this is one of the recent review, that is, you can see that nanoclusters have a diversified applications, including in the field of the sensing, biosensing, and the imaging. Next slide, please. Okay, this is one of the example. You know that copper sulfate is a very cheap salt that is you can find in your laboratory also. Here we have taken and we have added two surface functionalizing agent, that is, one is your glutathione, and glutathione, it will reduce the copper into copper metals and functionalize on the surface, and another ligand we have functionalized on the surface, that is cyclodextrin, and that is functionalized on the surface. There is dual functionalized your copper nanoclusters having size less than 3 nanometers, and you can see that it showed a bright red emissions. And here we apply a supramolecular concept. The cavity of the cyclodextrin was used to encapsulate the molecules like vitamin B6 cofactor. So whenever we have added a vitamin B6 cofactor, it formed a inclusion complexation, and the red fluorescence changed to the green. And then when aluminum is added, then nanocluster come closer and the fluorescence enhancement is occurred. So with a single system, we can able to detect sequentially the vitamin B6 cofactor, that is pyridoxal 5-phosphate, and the aluminum. So here you can see that this is the your UV. There is no in the UV. UV is the best way to characterize that whether you get the nanocluster or not, because it will not show the plasmonic band. It will not show intense band in the absorption. Okay, but when you record the emission, you will get intense emissions. But in the UV, if you will get an intense absorption, then you have not obtained the ultra small nanoclusters. So once we obtain these nanoclusters, we react with added different vitamin B6 cofactor. Vitamin B6, that is PLP, only form a inclusion complexation. So that's why color is changed. You can see the spectral changes also. Next slide. Okay, this is some of the experiment, that is titration we have performed, that how the fluorescence properties of your nanoclusters change with the addition of the your pyridoxal 5-phosphate. That will form the inclusion complexation, and that inclusion complexation we have studied separately by performing some 1H experiment, 1H NMR experiment, and some your DFT study. And from the DFT study, and by calculating the non-covalent interaction NCI plot, we see that the pyridoxal PLP form a good complex host gas inclusion complex with the cyclorelastine. That's why it's functionalized on the surface inside the cavity and change the your emission property of the nanocluster. Next. And using that PLP functionalized nanocluster, when we add the different metal ions, then addition of aluminum enhance the emissions. Enhance the emission because that because of the complexation occurred on the occurred between the nanocluster come closer, and that aggregation enhance the emission. Next slide. Okay, and that aggregation, that aluminum caused the aggregation of that nanocluster. That was we have characterized by performing the DLS. DLS state the change in the your diameters of that nanocluster. That change is because of the your that complexation induced aggregation of the nanoclusters. Next. So we applied, we did the titration experiment and determined the limit of detections. You know that limit of detection in the sensing field, generally we determined by 3 alpha by slope or 3.3 sometime. Alpha is the, you can take the blank samples. Whatever the you have the probe, you can take the 10 sample, and where you are going to determine the LOD, at that time you at that point you determine the your standard deviation of that blank and divided by slope of this calibration curve. Okay, that is the IUPAC approved equation to determine the limit of detections of a probe. So if you are new in the field of the sensing, so you can apply this equation for determine the LOD. Okay, if you anybody face problem, that you can contact me, or your the host, he is also working good in the field of the sensing also. Next slide. Okay, instead of these small molecules, maybe glutathione, that is tripeptides, you can go for the large proteins, proteins also. For example, BSA, LISO, HSA, different kind of proteins also you can use to prepare the nanocluster. Here we have prepared a gold nanocluster by lysozyme. Okay, it gives a rad emissions, and when you added different vitamin B6 cofactor, your pyridoxal and PLP, it has a aldehyde group. They form your you can say form a imine linkage with the pre-amine group present on the lysozyme, and alter the fluid emission properties of your of the uh, these nanoclusters. You can see the scheme. It will be easily you can understand. I have shown these different cofactor also. Only two cofactor change the your emission properties. That is here you can see the pyridoxal 5-phosphate. Okay, and when we added zinc in this, the nanocluster come closer, and we get a green fluorescence. So from red addition of PLP, it changes to yellow, and addition of zinc changes to green. You just see this mimicking the traffic light signals. You can by changing the input, you can change by keeping the input, you can change the color from red to yellow, yellow to green. Same nanocluster giving the multicolor emissions. And with this we have detect the zinc in the live cells also, in the and by bioimaging applications. And this is one of the application we have obtained from this lysozyme stabilized gold nanoclusters. Next slide, please. Okay, so we have applied this nanocluster for the developing bioassay, or you can say that probe for the detecting the alkaline phosphatase. Okay, alkaline phosphatase is it's an enzyme that helps or that catalyze the dephosphorylation reaction in the biological system. Okay, so it breaks the phosphate linkage. So that's why ALP activity determination is used for biomarker for diagnosis of various kind of disease. So and in the biological system, this ALP convert pyridoxal 5-phosphate into pyridoxal. So using this concept, we have tried to develop some nanocluster based probe to detect the ALP activity. Next slide, please. Here you can see we have used the same lyso AUNC, and then we have connected the your PLP, and it changed the color to the yellow. Whenever we added ALP, this ALP it cleaves the phosphate linkage and convert the PLP into pyridoxal, and change of this pyridoxal conversion of PLP into pyridoxal change the emission property of the nanocluster. So in that way we can able to detect the alkaline phosphatase activity. So I'm just repeating one more time that ALP is the enzyme that break the phosphate linkage. So we use the nanocluster as a light emitting unit, and the PLP, that is pyridoxal 5-phosphate, which is linked to this nanocluster by imine linkage, that is used as a phosphate substrate, where the ALP break the phosphate linkage and convert into the pyridoxal. Because of that conversion, the nanocluster properties change, and we can see that there is a clear change in the presence of ALP, and we can detect the ALP activity. Next slide, please. Okay, so we perform a titration also. Titration means we have added different amount of ALP ALP amount, that protein you have added. So different amount of ALP when added, titration showed a very good calibration curve, and which you can use for the analytical purpose also. And simultaneously, when the ALP is added, the fluorescent color is also changed. So that fluorescent color change we have recorded by the smartphone, that what is the change in the color intensity, and that using that smartphone also we develop a calibration curve and try to detect the alkaline phosphatase activity using the smartphone. Suppose you don't have a fluorescence spectrophotometer, you can calibrate the color change with the smartphone, and you can go for the detection. Next slide, please. Next slide, please. Okay, and whether the smartphone and the fluorescence by using the fluorimeter, both the approach we have detected the ALP activity in the real sample, and you see that the outcomes that both the methods are ideally good, and satisfactory results we have obtained to determine the alkaline phosphatase in the real samples. Next slide. So another observation we have obtained, that when the PLP is converted to pyridoxal, that time that time the emission changed toward the white light region. So we try to optimize the condition to add condition of that PLP and the pyridoxal, and try to develop the white light emission from the single nanocluster. And there is a very good important uses of white light emissions from the if you are obtaining from a single substance, single nanoparticle, or maybe single probe. So that can be used for different kind of optical devices. So here, when in the nanocluster, when you have added the optimized quantity of pyridoxal and the PLP, then we obtained a pure white light emission from the same lyso AUNC. Next slide, please. And we prepared, you see that nanocluster solution which emitting white light, and we prepared the gel, and we prepared some white light emitting LED bulb, as well as some we try to return some words on the papers. So so you see that same lyso AUNC, by using the vitamin B6 cofactors, we have find out a multiple application using the same nanocluster. Okay, next slide. Okay, so this is another applications. I will show number of examples, and I believe that you will be, if somebody get inspired and try to discuss or plan some more idea, or you want to enter in the field, then you are free to contact me anytime, no issue. Now I am in Odisha, so maybe I will back next month to Surat, then we can have a discussion, no issue. Okay, so here we have polyamines. You know that polyethylamines is there, that we have coated on the we prepared a copper nanoclusters. Okay, blue emitting nanoclusters. Oh, okay, here we have used the silver. Okay, silver nanocluster. And that amines is there, that amines we functionalize, or we make a imine linkage with the your pyridoxal 5-phosphate, that is PLP, and then this chip base form a complex with the your zinc and the cadmium, and you get a selective fluorescence enhancement. So this is a simple mechanism, that nanocluster is prepared using the silver nitrate by using the stabilizing agent PEI, and then PLP was functionalized through imine linkage, and it was applied for the sensing. And we prepare the paper strip and apply for the your sensing experiment. Next slide, please. Okay, these are some of the characterization by TAM, by DLS, and some of the titration experiment that and some of the spectral changes with different cofactors, and the spectral changes with the pyridoxal phosphate with the PEI stabilized silver nanoclusters. Next slide. So this is the analytical data showing the how this PEI functionalized silver nanoclusters get selective response in the presence of the your cadmium and zinc. Okay, next slide. Okay, next. Okay, so these are the titration to get the. Okay, the next slide, please. Okay, this is the most important things in this study I want to say, because mostly we go for the titrations, people and develop the sensors. It's very good, and they mostly people dip the when they develop the paper strip, they generally dip the papers into your sensor and they dry it. Okay, so when you apply, it may be you cannot reuse it. Okay, so what you did in this study, we chemically modified this paper, cellulose papers, by activating in NaOH solution. After activating, and then we use a divinyl sulfone, that is a linkage, and chemically we coat this paper with the nanoclusters, and then we characterize by XPS, that whether, and by the SAM. In the SAM pictures, you can see that in the last one, you can see that there are small dots are there. Okay, that dots nanoclusters that is coated on the your cellulose papers, and that chemically cellulose papers we have applied for the sensing field. Next. Okay, similarly, we applied the same similar approach in by preparing a copper nanoclusters, and we develop a chemically modified paper strip, and then apply for the detection of the picric acid. Here, the this is a copper sulfate, and by using the GSS stabilizing agent, we prepared the red emitting copper nanocluster. Those who are new to this field, I just want to say that it is a very simple approach to prepare all these nanoclusters. You don't need a very sophisticated facilities to develop all these nanoclusters, and in the field of sensing, you can you need not to extract the nanocluster also. You can directly use the in situ generated nanoclusters in the field of the your sensing also. Okay, so you see that copper sulfate is very cheap. Glutathione also you can get in very few hundred also, and then if you mix it in the presence of basic condition, in the basic condition you will get the red emitting nanoclusters. These are some of the characterization. No doubt getting the TAM image, good TAM image is little bit a time consuming, but only you need the TAM characterization of that nanocluster, and IR is very common instrument that you can use for the characterization. Next slide, please. Okay, so here you know that because of the addition of the different nitro compounds, the picric acid quench the fluorescence because of the energy transfer. Next slide, please. Okay, and that quenching, fluorescence quenching, we did the titration and find out the alloys of this picric acid. Next slide. And finally, here, this is the important things that we developed the chemically modified paper strip. Okay, in my talk, I discuss few new things, few maybe you know those who are doing the sensing, but few things that I discussed, that is, one is by use of the smartphone, and second, this one is the use of these chemically modified your paper strip. You can activate the cellulose paper strip, the paper strip that you are using in the laboratory. In the NaOH, take the NaOH solution, you activate it and preserve in the ethanol solution. That cellulose paper strip, that is you can react with the your nanocluster, and that functionalized molecule will be chemically coated on the surface. So if you repeated wash also, it will maintain the because it is chemically modified, so it will not wash away easily. You can use the repeated time. Okay, so here we have used and we have detect the picric acid by using this functionalized system. Okay, so finally, this may be my last slide. Sorry to all the participants that due to some internet problem, that talk was break in between. Okay, and finally, if you have any discussion, any questions, then we can discuss and you can ask me.
First of all, thank you, sir, for your nice presentation. I am very much impressed with your presentation because you have used the simple vitamin B6 cofactors, and then you have synthesized so many things, and that also used for in the practical purposes. So by using a single thing, you have developed the so many sensors and their practical purposes. That's why I'm very much impressed, because I am also started the work in this field, and I am using the carbohydrate as a what is called starting material basically. So
Yeah,
that's
yeah.
why, that's why I'm very much impressed in your presentation.
Yeah, it is a good practice because nowadays there may be sometime you will get fond or sometime you may not get fond.
And yes,
So
yes, yes, yes,
you think on the limited resources how good you can do. Okay,
yes,
and
yes.
second advantage is that if you concentrate on the similar one targeted field, you can go much deeper and you can contribute more chemistry to the society.
Yeah,
There
that
are
is also
two
good.
advantages.
Yeah. And sir, I can we use the any modified carbohydrates for synthesis of the nanocluster? Is it possible? Because
Yeah,
already,
because
every,
carbohydrate has so many functional group, OH functional group.
yeah.
Using carbohydrate also, you can prepare a number of nanoclusters because that carbohydrate will decorate on the surface and stabilize the nanoclusters.
Now, actually,
That is
sir, what should be the main strategy for design of the nano cluster? Actually, I have no idea about this nano, so I am that's why I'm asking.
yeah,
What should
mostly nanocluster if you are preparing generally NaOH in the basic conditions generally use. Okay,
okay, okay,
in that basic condition the reduction of nanocluster is takes place and that is the ideal condition, but there are some cases you have you have already added a reducing agent, then you can even you can prepare the nanocluster at lower pH also. But if you have not specifically not added any reducing agent and you if you want to go for the synthesis of nanocluster, then you can use some sodium hydroxide, hydrogens. Okay, these are some of the common reagents, common things that are generally used. Okay,
sir? Any specific functional group is required for these purposes?
generally these silver gold nanocluster they will prefer thiol groups to coat on the surface because of thiophilicity. Okay,
Okay,
but you can go for other functionality, OH also, OH, COH
I mean,
group, nitrogen amines group also. I given one example using the
yeah,
PEI, polyethylene. So these are some of the functional group that is that you can use.
okay, sir. If you give permission, then I can ask in the in future also. So, dear participants, if you have any questions, you can ask. Hello, hello. Am I audible?
Yes, you are clear.
Am I
Clear.
audible,
So,
sir?
uh, yes, yes, you can ask.
Am I audible, sir?
Yes, yes, audible, audible.
So I just wanted to ask that are these nanoclusters they are biocompatible or not? Are they biocompatible?
Yeah, actually, whenever you are using these are small clusters biocompatible, this is one of the more advantageous because these are you can prepare by using gold and the silver. Okay, but however, whenever mostly these are biocompatible, but if you are preparing new material, new nanocluster, you must check whether it is biocompatible or not. Okay, but most of the reports are saying that these nanoclusters are biocompatible in nature.
Okay, sir.
Hello, sir.
Yeah,
Okay, sir. Sir. Hello, sir.
Please ask.
yeah.
Sir, I wanted to ask, how can we calculate quantum yield?
Yeah, quantum yield, as I showed you that one is the absolute quantum yield, another is the relative quantum yield. So generally in the laboratory, we determine the relative quantum yield. Okay, that relative quantum yield you can determine by taking a standard. Okay, just like quinoline sulfate or fluorescein, whose quantum yield is already known. So you can record their spectra, UV spectra, including emission spectra, if you are recording in the same solvent. And then in the emission spectra, you just find out the area of that emission spectra, and there is a equation. That equation you can use to find out the relative that quantum yield. Okay, and whenever you are reporting such a quantum yield, you need to mention the which standard you are using. Okay, which standard and used for the determining that relative quantum yield.
Okay, sir. And sir, what is the difference between spacer and linker?
Spacer means link means if you have linked some with some group. Space means it's if you technically, that is a similar things, you can say that the emitted unit, that is light emitting unit, and the binding unit. Okay, that are separated by some group, maybe CS2. And I shown you two example. For example, that is pyrene CS2 NS2, and one is pyrene NH. And I directly connected with some chip base we have developed. But there is a CS2 group in another. Just let me simplify it. Spacer means it is separated by some kind of CS2, maybe some alkyl group, aryl group, which may not be conjugated. Okay, conjugation, there may not be conjugation from the light emitting unit and the binding site. Okay, that is, you can say that separated by some kind of space. Link means they are already linked together in the same system, integrated, you can say that.
Okay, sir. Thank you, sir.
Okay, any more? Sir, your mic is muted, huh?
Okay, sir. Sorry, sir. I think they have no questions, sir. Thank you for accepting our invitation and for your nice presentation, sir. Thank you.
Thank you to all the participants, and wish you a happy Diwali, and see you everyone in Surat, NIT Surat. Thank you,
Okay,
thank
sir.
you, thank
Okay,
you.
sir. Thank
So
you.
shall I go?
Yes, sir. Yeah, you can disconnect, sir.
Okay. Thank you very much.
Thank you, sir. So, dear participant, today session 3 has been completed. So next day we'll meet together again at 10 a.m. So all are